Hello, everyone, and welcome to your background on the machine learning project. So through this presentation, we're going to be going over just a little bit of what you guys had read and how this is relevant to how we're going to apply machine learning to understand symbionts and coral better. Okay, we'll get the laser pointer up right away. Let's go. Okay, first of all, this is a beautiful image from Madison Emery, and so all those little green bits are actually all the symbionts in there. This is actually a jellyfish, not what we are going to be studying, but they do host the same symbionts as our coral, and we'll get into that. So the evolutionary history of cnidarian symbiosis is kind of not super tricky, but it's just happened a lot over the course of Anthozoan and Medusozoan. Essentially, what this means is that these cnidarians can form an intracellular symbiosis with an algae. And this is common in cnidaria, and so all these green dots here represent ones that have an independent, at least one independent evolution of symbiosis. And essentially, it's just that classification of it houses the algal symbiont within the host itself, so whatever species you are looking at. So that's kind of what we're looking at from a big, broad standpoint of the phylum, but our corals are going to kind of hang out here. So as you guys read in your paper, you saw that in order for it to be established as symbiotic, it will lie in the gastrodermis layer, and essentially it gets housed in a vacuole. And while it is housed within that vacuole, the symbiotic relationship starts to occur. You have the symbiont providing nutrition to the coral host, and in turn, the coral host is providing a pretty nice home for that symbiont. And so that's actually, you can see it on the polyp here. So polyp is just like the little part of the animal for the coral, and that's where all its tentacles are. And then these are what they look like outside of the host. From the coral perspective, we actually know quite a bit about the algal symbiotic relationship with the host, in the sense of the host does take a hit in order to have the symbiotic relationship. So we know that the immune system is involved in the establishment and the maintenance of the symbiont population. There are quite a few papers that have gone into this to study it. Mostly like the PRR pathways or NF-kappa B will be suppressed. And this suppression may lead to some trade-offs, such as their immune system might be slightly compromised at this point. The other thing that's interesting about this is because with the immune system, some of these corals have actually evolved to kind of be exclusive in which symbiont they will house. So if it's not the correct symbiont that they want, they can just pop it right back out. And so this kind of is interesting because that kind of insinuates that there's something with the symbiont themselves that these corals have evolved to want for the symbiotic relationship to occur. And that may be dependent on which symbiont it is itself. So the overarching question for this project in particular is that based off of the things that we've kind of already discussed, so we know that corals have evolved to maintain this algal symbiont in their gastrodermis layer. We know that corals can evolve to be more selective with their symbionts, which means that they're selective in who will get established in the gastrodermis layer. Some other corals may have not been evolving in order to have a specific algal symbiotic partner. But the one thing that is drastically missing in all of this is, what is the symbiont doing? We know what products it provides because we know what's important to the host, but we don't know what the expression is in order to maintain the symbiotic relationship on the symbiont side. We don't even know if there is a difference for the symbiont if it's in one particular coral species or another. So that begs the big question to me of, does the gene expression of a symbiont change depending on the coral species it's housed in? And so that's where you guys come in. So meet our symbionts. Symbionts are normally classified by genera. In this study, you will be only looking at C, we'll call it C, but it's Cladocopium, the genera. But there's 4 major symbionts that kind of are housed in the Caribbean for the most part. We know this from ITS2 sequencing, so that is like a population study based off the ITS2 band, so we know which individuals are being housed. And that's how we know that there are certain species that prefer one symbiont over the other. So we have these 4 main guys. We have Symbiogeninium, Rivolium, Cladocopium, and Derstinium. And so some of these are examples, and we'll get to them in the coral, that are evolved to match to a specific, from the host side, the host has a specific partner it will want to choose. In particular, Rivolium and Cladocopium seem to be the ones that have the most diverse amount of species associated with it, and kind of the more popular symbionts overall. Derstinium is relatively new, and for a fun fact for you all, it's very exciting for a lot of Caribbean coral researchers or Florida coral researchers, because it is supported in some studies that Derstinium does have an increased heat tolerance. So when corals bleach, they're actually expelling these symbionts, and that can lead to starvation and death if the heat stress is maintained for a long period of time, like you might have seen with the Florida Reef Tract this summer. It's thought that if we can give more Derstinium out there, they might have a better heat tolerance overall, but that doesn't seem to be the case if the heat does exist for more than, 4 days. So here are our corals. So you guys have been divided into teams already. I'll hint towards that towards the end. But these are your corals that will be in the study. So we have M. cavernosa, Orbicella annularis, P. strigosa, and Parietes asteroides. So all of these are stony corals. So what that means is that they are kind of the big reef-building corals. These are the ones that look kind of like boulders or rocks. These are the ones that people have a hard time believing are animals. And essentially all these little things are the polyps, and this is where the symbionts will be housed. M. cavernosa can house B, C, and D. It doesn't seem to be very specific in which one that it normally houses, although it seems to favor C and B over anything else. Orbicella annularis, that one is closely related to M. cavernosa. That one will house primarily B and C. Can house D, doesn't normally house A. P. strigosa tends to favor B for the most part, and Parietes asteroides is very exclusive towards A. It doesn't really like to house anything else. It really only likes A. It doesn't mean it can't. It just means that it prefers that. So we've talked about how we have the coral host and we have the algal symbiont, and we want to know the gene expression of the algal symbiont. Well, you can't really bleach the symbiont out and then get the gene expression information. You have to sequence everything all at once from the host, which means you have to separate what is known to be a symbiont gene expression from a coral host expression. So what that means is that we have to use very specific bioinformatic tools in order to get that information. And so this is the pipeline that we use in our lab in order to be able to get that information. And so FASTP is used to trim, and then after everything is trimmed, we use this program called bbsplit, or this command bbsplit from the program bbmaps. And essentially what this does is it takes your sequence here, my beautiful graphic, and you give it either transcriptomes or genomes that you know are associated with the individuals that should be within this sample that you have. So then it'll look at each one of the contigs within your sample and sort them to which one it matches to the best. And so then you get, for our study, we have A, B, C, and D, the host, and then this kind of like dump file that like whatever didn't match everything else gets dumped in there. So we can look for viruses, we can look for other eukaryotic organisms, sometimes bacteria get in there. That could be a dump file, hangs out. And then from there, we can take each one of these samples that it got sorted out, use Salmon program in order to quantify our reads, and then we can use DE-seq because we use R in our lab to get the differential expression and normalize our samples, or gene counts, that is. For the purpose of this study, we're only going to be looking at C, because C tends to have the most reads associated with all the samples. So just to make it easier for you guys. So your task, which has already been assigned, is that each group will be tasked with answering a slightly different question using some of the methods that were discussed in the background that I gave prior. Each group will be attempting to classify cladocopians, so clade C, based on which host it's found in. So you could be assigned to MCAV, PAST, PSTRIG, OAN. So each group are going to need to classify their questions. So MCAV to the gene expression of the symbiont using the following algorithms. We're going to use logistic regression, linear discriminant analysis, quadratic discriminant analysis, support vector machine learning, and then partial least squares. Essentially, what is going to happen here is that over the course of you guys working together, you will be able to look at all 5, and you will have this final product of a group presentation, a group report, the satisfaction of learning machine learning protocols for genetic expression research, and hopefully some fun tidbits about coral along the way. In addition to this, each of your groups will be looking at each of these 5 algorithms and determining which one is classifying the information of the genetic expression best to your host. I can't guarantee which one it's going to be. It might be one for one species and it might be one for another species. But that's the fun of it. Machine learning isn't always a one-size-fits-all, and it's important to realize that you might have to test a couple different algorithms in order to best understand your data. So for best practices for this, you should be watching and walking through the tutorials that I will provide. You should be talking with your groups. If you also look, there might be other groups that are doing the same species as you. You can check in with them too. That might be helpful. And of course, you can email me with questions. Preferably email Jeff if it's a Python question, because your girl is a R gal. So that I can try my best, but I am always open to talking about machine learning in general. So that is kind of it for the project. Please make sure that you are following up with the tutorial. Make sure that you are utilizing the resources that I have linked in the module, so that way you guys don't feel lost. And if you do feel lost at all, just make sure that you take the time to email me, and I will make sure we will get you where you need to go. Okay, looking forward to seeing all your projects.